Okay, good morning, everyone. And welcome to the last day of the lectures. We'll start with the first talk, which will be given by David Kouřil. David Kouřil is a postdoc researcher here with us at Masaryk University. He will be talking about navigating and making sense of large 3D visualization, which I think is partly about what he was doing during his PhD research at TU Wien. And he actually received a Ruiz Award for his dissertation thesis. So I think you are in for some exciting things. Thank you, David, for agreeing to give us the talk, and the floor is yours.
Okay, hello. So this works, the microphone. Cool. So thank you for coming in this morning. I was asked to give a talk basically about what I was doing in a PhD, as Katka was saying, and sorry that I couldn't have been part of the early sessions because I'm having currently a almost 2-week-old daughter, a newborn. So unfortunately, I couldn't hear the great talks. I saw that you had quite some excellent speakers. So yeah, I'm going to be just basically telling you what I did for a PhD, which was already like a 1 year plus ago. And then I'm also, if there's time, going to tell you a little bit about what we're doing here currently, what is the project that we are working here with some PhD students and some more senior researchers. And the topic, yeah, it's about navigating and making sense of large 3D visualizations. And I'm going to have to a little bit improvise because I don't have a presenter view here. So who knows what's the next slide? Oh, yeah. So I just wanted to say, like Katka basically said it, I'm postdoc here currently. I did my PhD in Vienna, which some of you are also familiar with. And I did my undergraduate here also. Here at Masaryk, I was at some point really into this generative design, creative coding, but then I met this one French guy who was an excellent graphics programmer who was doing a PhD also in Vienna, and he kind of lured me into this world of scientific visualization and working with biological data. So I did my master thesis in Vienna already and kind of started in this topic. And this subtitle that I call the talk is "You visualized it, but now what?" And what I wanted to express with that is that a lot of the times the visualization researchers are involved in how to render data, how to put the data points on a screen. But I think the most important and interesting part, at least for me, is what happens afterwards. After you have a renderer that is able to render something that's a data set that's quite large, and you want to enable people to interact with it and navigate it. And I was most specifically working with biological data, but this is kind of overreaching problem, I think, that we are tackling currently. So what can you imagine as a 3D, like spatial data? I have these 3 examples. So most of you are pretty familiar, I would guess, with something like Google Maps, like online map services. And I don't know if you've ever thought about what goes into actually making this possible, how you can, with such a simple gesture, with such an intuitive interaction, go from some place like Brno to Manhattan. And there's research behind this, research in human-computer interaction. And for me, it's just amazing how such a large-scale object, an Earth, like the surface of our Earth, how it can be represented in a computer and navigated in such a simple, seamless interaction. So if you think about it further, these days we are also able to capture something that's beyond just the surface area. We can use some laser scanning to get point clouds of 3D structures. And there are methods how to get city scale scans. And again, you can think about how would you navigate it. Like computer games have these metaphors of walking, but it's clearly not scalable. If you want to walk from Brno to Manhattan in a virtual environment, you cannot really do this. And then the final example is what I've been involved, which is biological data. And I don't know if you see the mental step that you need to take from the second to the third picture, because here you still have a definition of a surface. You know where is the plane that you can walk on, but here, this is in all 3 dimensions occupied by objects, and there's no orientation of up and down. So that makes navigation and interaction more difficult. And that's basically became the topic of my research during my PhD. And I was working, as I said, specifically with biological data. And in visualization, this is a very rich field. Sometimes molecular graphics is mentioned as one of the earliest examples of scientific visualization. So this is basically not even a system, but from 66 people were able to put images, rotating images of molecules on a computer screen, and this was quite revolutionary for chemistry, because suddenly people were able to really see the spatial characteristics of the objects that they are dealing with. And these days, of course, before this was an amazing success to just show one single protein, but these days we are going beyond. So we can image very much larger proteins, even viruses, and sometimes even cells. So this is like a 100 times magnitude of scale increased in the models that you have to represent and navigate. And computer graphics in general, thanks to modern GPUs, we are able to render these models interactively. So now really the problem is not how to render, how to display these data points, but how to navigate them. And just to illustrate, here's a video of what was the status basically in 2015 where I came in. So people were able, this is in Unity, they were able to render, I think this is like billions of atoms in real time. And of course, there's like a level of detail schemes and some smart techniques so that you don't always render the billions of atoms. But what you can see is that at this level of scale, the atom is not even the lowest granularity that you see on a screen. And this problem of scale and difference in scales between different biological objects, it's quite interesting to me. So I was doing some tests during my studies with like what was on the left was a size of a cell compared to a size of a virus. This is HIV. And you can see that even just like the simple scrolling interaction that you would say is quite natural, it has its flaws because you have to traverse quite a large amount of space to jump between these two sizes. So that's really the issue, that we have multi-scale data, and in biology this is complemented by other, let's say, complicating characteristics. Like you have these multiple instances, you have several types of objects that are then represented thousands or millions of times in the scene. And these models are hierarchical. So, you know, proteins build protein complexes, and these build viruses, bacteria, and cells, and other like cell organelles. So there's a hierarchical relationship. And as you can see, this data is quite dense. It's almost like a volumetric, like a slab of volumetric data, and that has to be dealt with. And it's what we call truly 3D, like I already kind of mentioned. You have detail or distributed in all 3 directions. And the question again is how to enable people to interact with. And through the presentation, there are like these 2 red threads, also through the papers that I was doing. And that's not only enabling interacting scientists and biologists with their data, but also participating in what's called science outreach, which is using kind of visual methods for communicating science to the general public. So that's one of the topics that the red threads that I was dealing with. And the other, as already kind of mentioned, is exploring these scales, these magnitudes of scales. Maybe if you haven't heard this term, science outreach, it's, as I said, it's this communicating scientific concepts to people who are not trained as scientists. And but it doesn't mean necessarily that it only helps or it only entertains people. It also is very useful for the science, for the scientists themselves, because it helps put things into perspective. It enables people to hypothesize. And one of the things is that there's like a saying that if you want to know something, how to do something right, or what is the truth, you just write on the internet what is something that's not correct, and people are going to come and correct you. So this works also in a similar way here. If somebody makes an animation and a biologist sees it and realizes that it's wrong, that they're going to come in, and this way they can kind of iterate on it. And it's sometimes very cool looking piece of art, and there's no better example than David Kouřil. So these are hand painted paintings, not illustrations, but paintings of cellular environments. And this was, again, hugely influential because it bridged the gap between molecular scale, just looking at single proteins and molecules that are quite small, let's say, and looking into larger organisms like cells or bacteria. And the other example is Drew Berry, who's using Maya to make these animations, and it's quite amazing. So you can look it up if this is something that you're interested in. And for me, like the one I always like to show this inspiration, this game called Everything, where you could basically play as everything. So you start at a human scale, and then you can kind of jump into a squirrel, and then you can jump into, I don't know, even a smaller animal, and then into bacteria and some pollen, and you go down into DNA. And this is quite influential to me. This is quite something that I would like to have, but with real biological data. And this is quite a distant goal, but I always like to remind this. Yeah, so, you know, when you're doing a PhD, you're doing papers. So I had these 3 papers, and the connecting topic is, again, navigation. And navigation generally in 3D graphics is quite extensively researched, but there's one major flaw, and that's what I already mentioned, that they always assume that you're doing something that has a planar surface. Most of the time, this is what this research is focused on. But then for us, if we are doing multi-scale biological environments, this just has such a unique characteristics that need novel approaches. Yeah, and again, another influential piece of media. I don't know if you've ever seen this. This is called Powers of 10. It's a video, again, from quite a distant time, from the 77. And it has these 2 parts. So first of all, it starts at this human scale and zooms out to the end of the observable universe. And then it comes back and goes on the what we call negative scales, looks inside the person lying on the grass. And what you can maybe observe is the density of detail that is much higher on the negative scales. But this is like such an influential piece of media that, again, helps to put things into perspective, and you realize the big picture. So for me, visualization, data visualization, and scientific visualization is just another medium that is so suitable for science communication. And so that's kind of what I dedicated the research for. And interaction with visualization for general public, for people that are not trained, maybe not even that well versed in computers, it requires very intuitive interaction methods. And I guess you could argue that the most easiest interaction is when there is no interaction at all. So if you have a movie and you don't have to decide what's gonna happen in that movie, and you're just presented a story, this is the most easiest that you can interact with it, because you're not interacting at all. And so there is this range between fully interactive applications where you have to control everything, and then between something like a pre-rendered animation that just shows you something. And for us in visualization, this is the difference between explanation and exploration. And I'm not the first one to realize that you can actually achieve both with data visualization. So Anders Schindemann, he coined his name, exploration, that basically expresses how in visualization system you can do both. You can both explain something that you want, some storyline or some fact, and you can let user explore. So I'm really heavily leaning on my thesis. So I was presenting that you have this interaction spectrum, and on one side you have some examples like computer games, on the other you have some movies or books, and then it's not just one or the other, but there's a range in between. So people have been and like the more you go from one side, you have more ability to storytell, and from the other side you have more ability for users to choose what they want to see. And there has been different means of dividing this spectrum. So people have been looking at guidance. Let's say you're guiding the user, then this work is more on the specifically for scientific visualization, I believe. And in my thesis, I basically defined navigational modes that go again from this. You have to control everything to you control nothing. And I was calling it navigational modes. You have this augmentative navigation, declarative navigation, and automative navigation. Right. And with navigation, this might not be so obvious, but the first step for me was textual labels. And again, coming back to the example of online maps, can you tell what city this is? Like maybe I don't see anybody who's Austrian, so okay. So maybe you could, from the shape of the river or the shape of the city in general, you could infer that this is Vienna. But without it, when you don't know it, it's so hard to know what you're looking at, and how can you explore and navigate an environment when you don't even know what you're looking at. And in molecular and biological visualization, we often have the same problem. Like the tools are developed for scientists, and they maybe spend their whole 50-year career studying one protein, so they know everything about it. But if you show it to a person that has never seen it, that maybe had elementary school biology, then they need some guidance, they need some annotation. And so that's basically what my first work was, to get labeling on these biological scenes, on these multi-scale biological scenes. And this is what it kind of looks like. So you suddenly don't only see some red stuff there, but you know that this is blood plasma. And if you zoom in, you can see what kind of a blood protein this is. And I hope I'm not going to run over time, but I'm going to quickly explain how this method works. So again, labeling in computer graphics is such a well-researched area, but here we have these specific characteristics that make it harder, and you cannot use the method that exists. And it kind of works in two steps or 3 steps. We have the multi-scale step, in which we are deciding on what level each object is going to be labeled. So we basically divide the scene based on the Z coordinate, and then the higher the further from camera a certain object is, the higher on the level of hierarchy is going to be labeled. And this is basically a similar idea from normal computer graphics for geometry. So level of detail techniques work by kind of degrading the geometry, and this is very similar idea. You're lessening the detail of description. So it's LOD applied for labels. And yes, we did choose the name of this technique, not arbitrarily. Yeah, so basically that's what I mentioned. This is how it works in multi-scale step. So you kind of segment the scene based on the distance from camera. And in the multi-instance step, you need to, as I mentioned, like this data is multi-instance. So you have one protein that is there, then maybe in the scene like thousands of times. So you need to pick which one of these instances you're going to label. There are some criteria, and we combine them together and basically pick the one with the best score. So we choose the instances, and then in the final labeling step, we place the labels. And there are, in principle, these 2 types. So you either overlay, which is called internal labels, or you shift them a little bit away from the object that is labeled, and you connect that with a lead line and an anchor point. And this is called external labeling. So we are using internal labels for 3 reasons. And this is what we end up with if we don't consider temporal coherence. So we are making something that always makes a computation each frame, and this results in a very stable, very unstable behavior across several frames if you're making some movements with camera. So you need to consider temporal coherence. We have made some adjustments, some mechanisms of ensuring temporal coherence, and this is what it looks like when you do that. So it's again making it maybe more smoother for the user. Yes, basically the labels are staying upon interaction, and they are only updating once the camera stops, and it also there's this kind of nice transition. So I think that's like summarize the first work. This was really the first step of including some sort of annotation, some sort of guidance for a user so that they can navigate the visualization better, or even enable to navigate them at all. And what this does is that it allows better inquiry. So if we've had these like researchers nights, and people are coming in and we show them this nice image of HIV in blood plasma, and then they look and they have no idea what to ask because they don't know what they're looking at. So now if they have labels, they can come in, and instead of asking like, okay, so what is this like red thing inside? They can maybe ask rather, okay, what does RNA, which is the red thing inside, do, and how significant it is for the virus? So that was kind of my contribution towards the augmentative navigation, where the user still deals with everything, but they are getting some pointers to where to look at and what they are looking at. The second work was something that goes more towards automating the navigation process itself. And the motivation for this is that we have these biological structures that have interesting detail on many levels and on many kind of ways of how you can cut them. But if you simply show the data set themselves in full, you don't get to see this nice detail. So there's the particle, the HIV particle that was shown there before, it's embedded inside this blood plasma, but you cannot see it if you don't do some sort of cutting away. So how you could explore this data. So maybe you have a list of all the types of objects, and you click on the list and it shows the object. But then this is completely separated from the hierarchy and how things are put together. And you could make this into some sort of a tree view, but then if you have 100 objects types, this is just, you don't have enough screen space. So to support people to making this sense of how things are put together in biological objects, we made a method that allows people to hierarchically browse. That's the keyword, the 3D model. And browsing, this name is not taken again arbitrarily, but it is used because we are basically using the same metaphor as you are using in web. So in web, you are also browsing, you're clicking on elements, you're clicking on links that are taking you to different locations. And on the web, it might not seem like such a hierarchical organization. So another example is in file system. And again, these browsers have a hierarchical organization of folders, and you are traversing this quite large information space by clicking on elements, and you're doing this stepwise steps. You know, you have in Finder, you always have this option of switching directly into a folder that you want, into directory you want, but you never use it. Even if it means that you have to jump back, and then go back down, you're going to do that because that's much more intuitive for you than typing. Maybe if we are not talking about terminal use. So yeah, so this method, we call it hyperlabels again, and kind of similar to hyperlinks to kind of drive in on the metaphor. And what the technique is about is that you still have the labels that are giving you the orientation, but you can now click on them and you basically dive into that object, and it's revealed to you, and you see what maybe the virus is made out of. There's another video. So it basically enables you really quickly to navigate this large, both hierarchical organized information space and also the 3D spatial, this spatial space, if you say. The method itself, it basically has these three major elements: the hyperlabel, that is basically an active annotation element, the breadcrumbs panel, and the structure of interest opening, which is just like the means of opening up the structure and how it is done, how it is defined. So the hyperlabels, they allow you to drill down. So you start at the highest level, you show the whole model, and you click on the subparts that you want to explore, and you go down. But then if you get to the lowest level, how do you go up? So again, we use a technique from web design. This was hugely popular when I was starting to doing web design, maybe in like 2007, 2010. So we had these breadcrumbs panels. And Amazon still uses it, and some e-shops still use it, not so much web pages these days. So this breadcrumbs panel that we use in the hyperlabels technique is the same concept. You have the path from the top to where you currently are, and you can click on any of these, and it takes you back to that location. And a structure of interest opening is just a fancy way of saying that you have to deal with somehow the occlusion. So you have to make some cutaways in a smart way so that it considers the semantics of what you clicked at. There are certain ways how you can adjust the visibility. So for example, if you only show one type of an object that is like a child of what you clicked on, then you lose some spatial context. Like you lose the idea that there is this spherical membrane. So you might just have a different design for keeping this context. And there's a different visual style that you can employ, because sometimes biologists, they can get very confused. If you make a cutaway and hide some objects, they're going to be like, oh, where did it go? What's wrong? Why is it not there? So you can also experiment with how you indicate the parts that you're cutting away. And you have to change the coordinate system of the objects that you're looking at. And this is what actually does the traversing of large distances in space. And you have to, in the final step, you have to deploy new labels on the new level, update the breadcrumbs panel. Yeah, so this is basically how it works. And yeah, it might not seem from it, because I'm doing like a really quick overview of this technique, but I actually quite like this paper. I think this is the best work that we did, and I like it because it doesn't seem so complicated. You look at it and you're like, oh, okay, well, this is obvious. Like, what's the research about this? But oftentimes this is where you actually know that things are working very well. If somebody comes to your visualization system and then it's so unobstructive, so intuitive that they can pick it up quite quickly, that's kind of a sign that you design things well. And yeah, I mentioned that this is done to support this stepwise navigation, not just jumping to some arbitrary random access objects. All right, so then the third one, this is the mode where the user basically doesn't have to do anything. They are just presented a story that somebody maybe authored, and they don't have to deal with how to set up the cutting plane, how to navigate the camera, but they just see something interesting on screen. And for this, my contribution is a technique called molecumentary, which is molecular documentary. So we generate these guided tours of these large-scale, multi-scale molecular objects models. I don't think there's now this sound, but basically the idea is that if you show again this to user, they are not going to be able to get any insight from this. They look at it and they're like, oh, okay, that's a nice picture, but they're not going to learn about the functionality of all these objects. And this is quite shame, because this is at least for me and many others, this is quite a fascinating field and quite fascinating thing to learn about how life works on these microscopic scales. And there's many very engaging explanations, maybe in a form of a book, or even if you go to Wikipedia or some other materials, this is quite nice to read about. But then the visual part is often very neglected because, for one, in books, you never can see dynamics, you never can see animations. So as a counterpoint to that, we also have these scientific videos. Like these days, many even students are looking at if they want to learn something when they are studying biology, they go on YouTube and they look for some animation that somebody made, and this is how they learn. But the problem here is that these animations, they are quite they take a lot of time to create. And once, let's say, biology is very much changing all the time. So let's say you spend 6 months making an animation, and then somebody comes in with a new insight, and then you have to redo it. This is not scalable. So what we were proposing, something how to make this documentary creation more scalable. So instead of using some animation software that outputs you animation in like 30 minutes, let's say 1 hour, you just use real-time visualization that gives you the images in real time. And instead of altering manually the animations, the transitions, we use some sort of automated camera exploration. And instead of pre-recording a voiceover that explains what's happening on a screen, we are using synthetic commentary using text-to-speech technology. So there's a whole overview of the framework. I'm really not going to go into the detail, but basically what it does is it's making these scenes based on what objects are in focus, and it's using some procedural camera animations and using the cutting plane to dynamically reveal what you're looking at. And the interesting part, at least in this work, is the usage of audio, because in visualization we are often not very fond of making data sonified, making them into sound, because sound has an inherent flaw as opposed to visualization. If you look at an image, you can get an overview in a blink of a second, but if you're listening to sound, you have to listen to the whole thing if you want to get a glimpse or get an overview. But in this work, it quite fits. So we are using text-to-speech to basically get some text explanations about the objects in the model and then deploy it on demand. So somebody, you look at a protein, you look at an albumin, and then the text tells you, the voiceover that is generated tells you what albumin is for. And there are two basically types of what you can do with this technology. So one is that you can basically have an endless loop of exploring the model in a scenario, let's say in a museum, or if somebody comes to a science center, and then the model is always being explained and its parts. So that's one scenario of using this. And the other is text to mockumentary. And I'm going to try to do this sound, so I have to not speak.
Researchers are studying GP120 in the search for new approaches to creating an anti-HIV vaccine. Several viral enzymes are packaged inside the virus. Reverse transcriptase will create a DNA copy of the genome when the virus infects a cell. Integrase will splice this DNA copy into the cell's own genome, which then forces the cell to make many.
Yeah, so basically what you saw on the left side there is a text that is written by a biologist, and it explains how the virus works. And then we take it, we cut the sentences and find out what are the objects of interest that are being told, and we automatically show them. And you heard the voiceover. These days it's not as nice as if a person does it, but it's quite believable already, and it's not like the Stephen Hawking robotic voice anymore. So that was my example of doing visualization that is automated, that is, you don't have to do anything to explore a visualization. It just reveals the model itself to you. And as I mentioned, this is for visualization, at least. I think it's quite interesting how we leverage the visual, the audio track. Right, so that's basically finishing the part of the PhD, and to kind of end up, I showed you this Powers of 10 video, and the goal really is some distant goal, maybe in 20 years, to be able to integrate data sources and then be able to make something like the video, but interactive, where you can basically, on any scale, you can explore, dive in, dive out, and learn about how life works in this interactive manner. And if I have just like few minutes, 5 to 10 minutes, I wanted to also mention the work that we are doing now here at Masaryk where I'm a postdoc. So we have a team of people, like 2 PhD students and some senior researchers. And we are looking at chromatin visualization. And if you know what chromatin is, it's basically a name for DNA that is wrapped up in cell nucleus. So you probably from your biology class, you know that the DNA is made by these nucleotides. But the interesting thing is that a DNA in each cell in your human body, the DNA is almost 2 meters long, like the real distance if you laid it out. But then the nucleus diameter is something like 10 microns. And the thing is, like, how the hell is it compacted in a way that a 2-meter thing is packed into this small nucleus? And I was thinking, we have the researchers night coming up, and I was thinking how to better have people to visualize this this size difference. And I was thinking, you know, the Kinder Surprise, this capsule, you have this chocolate egg and the capsule inside. So if you wanted to represent that as the cell nucleus, the length of DNA, it would be something like from Brno. Oh yeah, you are most likely not all from Brno. So let's say the distance between Prague and Brno, maybe something like 1/4 of that. You have to pack this into the Kinder Surprise capsule. So this is something like quite amazing how nature does it, and people are looking at how this is done, and they know that there are certain proteins that are helping to compact this DNA, and there's this hierarchical organization of the DNA being wrapped around histones, and then these histones and the DNA together called nucleosomes, and this again is compacted into a fiber, and this is compacted into the cell itself. So yeah, this is just another image of it. And the problem with looking at this structure is again that it's multi-scale. So you can look at at the cell with a microscope, but then you cannot with a microscope see the DNA itself. There's just a discrepancy between resolutions and size scale that you can capture with each resolution technique. So the multi-scale aspect is always problematic. But biologists came up how to learn at least some information about how the chromatin fiber is wound up in the cell nucleus. It's called Hi-C. What it basically does, it fixes the parts of chromatin that are close together, and then there's some cutting and some sequencing, and this way they can learn somehow what parts of DNA are in close proximity, in physical close proximity. The output is are these Hi-C maps. This looks quite like something maybe, you know, adjacency matrices or some heat maps from other fields of computer graphics. And believe it or not, but biologists can argue and can learn some insights just from this very confusing representation. But it might take one whole PhD to be able to do that. So for us as visualization people, we are again more interested in what this actually looks like in 3D, and there are ways how to use these Hi-C matrices to simulate what chromatin could look like in the 3D conformation. So we have these 3D models that are a prediction of chromatin, and again, already from this prediction, you can make some insights, like these colors are, I believe, chromosomes, and you can look at, for example, the yellow chromosome is kind of like with this shape and it's close to the purple chromosome, and that might be of significance to biologists already. Yeah, so, and maybe why it's important to look at the 3D structure and how things are closed in 3D space is that you have these genes and other regulatory elements on the DNA, and they can be quite distant. So let's say if you need one gene to activate, or one transcription factor to activate one gene, and they are quite far, you might think, like, how is it possible that they can make this travel? But the thing is that these might be quite far in a linear sequence, but they might be in space quite close by. So this is important for biologists. And there are some tools to do that, but the visual language, basically the visualization, it's far from being standardized how it is maybe for molecules. And if we look at it from like 3D visualization perspective, this is quite rudimentary. This is quite basic. This is maybe the computer graphics that you could make 10 years ago, but now we can make much nicer and much useful 3D renderings and interaction methods. So we have this tool. I'm almost finishing here. We have this tool that we are developing that should be useful, hopefully. And we, from the visualization perspective, maybe the kind of last point, it's interesting to look at different representations of one data, and think about what makes sense to do, what actions and interaction it makes sense to make in one representation and in another. So for us, it's look at the high-C map, the 2D representation, and look at the 3D, the structure prediction. So this is kind of a question for visualization research. And the last example of work that I did, so I was now doing, I'm now working with this, in this genomic field, let's say, genomic visualization. And even during my PhD, we worked on a project that was trying to emulate an illustration, something like I already showed before, or something like this, where you have shown the different scales of organization that is quite hard to capture on one single image. And we try to make it into an interactive system. So the interaction was quite important. And you can see, like, the idea was that you have these different panels or planes that are of different scale, and you can kind of scroll through.
We can pan any of the scales beyond the chromosome scale, which makes the remaining scales pan as well at the appropriate higher or lower speeds, depending on the scale difference.
And I didn't prepare any conclusion or lesson also learned, so thank you, and ask questions.
Okay, thank you, David, for a very, very nice talk. First, let me pass on congratulations from Sarah on Discord to your new family edition.
Thank you.
And now let's ask questions. Are there any questions for David? Yeah?
Sure.
First and foremost, thanks very much for the talk. It was amazing. And I was thinking that perhaps the label location, you chose it to be inside of the presented object, yes? Mm-hmm. Could it be that in some scenarios where the label might be a quite large description, could obstruct the intended object to be viewed? And is there actually a functionality in place to perhaps move that label by the user? Because in some cases it might be the case where, for instance, I would like to see a protein on a close-up basis to see how it's folded, and perhaps the name is just in the way.
Mm-hmm. Yeah, totally. I mean, that's definitely something. It's just one design, how you could use it, this is basically designed for the way that a user approaches, and they want to see what they're looking at. They want to make a sense of it. So definitely, if you want to get some, let's say, longer descriptions, you would use something else. You would use maybe some external labels where the object is unobscured, as you mentioned, and you have the textual annotation or some other information around it. So it's definitely something that could be considered a design for this scenario. So we are always like in visualization thinking in terms of what is good for this use case. And here it wasn't like that you have a specific task that you need to, okay, I need to locate this one and then I need to learn something about it. It was really more of an exploration and kind of concept in place. But yeah, great question.
And regarding the exploration aspect, this might be not really geared towards the unexperienced user, but more perhaps to the aspiring students in the field of medicine and biology. The exploration process with the breadcrumbs is very intuitive to some people, like you said, and is still used in this day and age. But I was thinking that perhaps a quote-unquote incision tool where a substrate might be cut away would also be intuitive for, let's say, medicine students.
Mm-hmm. Mm-hmm. Yeah, sure. Sounds, yeah.
Thanks.
Nothing to comment. Yeah, good ideas.
I think there was one more question.
You had a question, right?
Yeah, actually, my question was the same as his first. But going on to internal and external labeling, is it correct that you didn't choose external labeling because the line would just vanish if you zoom too much?
No, why we didn't use the external labeling was mostly because here, for external labeling, you need a definition of an empty space. So it's most often used where you have a very external view. Let's say in medical, it was used, you have a model of a human, and then you're looking at it like from distance, and then you can externally label like, okay, this is a liver, this is a heart. But here we can dive in wherever you're surrounded in the model, in molecules. So then it's kind of maybe harder to define what is an empty space. It could be done where you could place the external labeling, but the reason why we didn't choose external labels is this, because you don't have an empty space basically. And also the thing is, what we wanted to do, so we have these objects that are physical objects in a scene, in these proteins. And then we are labeling also groups of proteins. So let's say the plasma as an example. Like if we would use external labels, then you have this anchor point that ends at a specific point, specific object. So that could be it could be ambiguous, because you wouldn't know, the user wouldn't know if you're pointing specifically to this one object or this bigger group. So we kind of like this, that you kind of don't point to any specific point, any specific objects with the internal labels, and to use that.
I see. Thank you.
Yeah, thank you for your questions. Are there any more questions? Yeah, there is one more.
The last thing that you showed, it was DNA, right?
Which one? Oh, you mean the
one where you can scroll?
This one?
Yes.
Mm-hmm. Yeah.
Is this just some random visualization showing that, or is this like a real DNA that you can, for example, overlap? This part is this gene, for example, and then an option to zoom in or how it is rolled onto, for example.
Yeah, so I'm not sure about the base pairs. Like the base pairs are the only thing that is the data that you could have. So I'm not sure if this was mainly driven by a PhD student that I was like co-advising. I'm not sure if he used a specific sequence from like a sequence, the human DNA, let's say, but really anything beyond that, we cannot really say how it is. Like even the sequencing of human DNA, it was like a huge undertaking, putting together like all these different data sources or experiments. So I think in this case, there was some data again coming from Hi-C, like the structure prediction for maybe, I think the nucleotides, this loop thinking. But then again, we are kind of bending it into this shape from the spatial conformation to straightening it out. So this information is a little bit lost. So, I mean, it's good thinking and kind of would be interesting future work to really use this to explore the sequence. Yeah.
Yeah, I was thinking like this to become like a real map of the human genome.
Yeah.
Where you can explore where are which genes, how they're correlated, which part are unknown and all of that. So.
Yeah, that would be amazing. Okay.
Thank you.
Thank you so much.
Thank you. Any more questions for David? If not, then let's thank him once more.